We're going to talk about organelles and cell structures. And you can write this down right now. Those of you who haven't been in my class, the way I do it is when I give you notes like this, like from a slideshow, PowerPoint slideshow, write down the stuff that I underline. If you want to write down more, knock yourself out, but you don't have to. The important stuff will get underlined. And if I add stuff, like if I add a diagram or if I add words, write that down too. But you do not have to write down every word that appears on every slide. You never be able to. You can't keep up, all right? So today, we're going to be looking at organelles and cell structures. And again, those of you who are new, I'm going to kind of lay out for you what I'm expecting, and I'm going to lay it out for you in a way that I hope will help guide how you approach the work that we do today. Okay, so here's what I want you guys to think about. First of all, we're going to do a quick review of cells, what they are and why they're important. And I want you all to understand that. That's mostly going to be me, though. I'm just going to be talking. And then I'm going to talk some more. And the talking here is going to be aimed toward getting you to recognize some stuff. You're not going to know it. You're not going to remember it. You don't have to memorize it. And I'm not even going to ask you to write it down. Instead, I want you to have an initial, like, oh, okay, there's this. Oh, there's this. Oh, yeah, that word. Okay. And what happens, the way that people learn, the way that our brains work, is it's like building a structure. There's this foundational piece that you don't necessarily see, but it's really important. Whatever's on the bottom of the structure is invisible, but if it weren't there, the other stuff couldn't be put on top of it. That's the piece that I'm going to do today, some foundational work. And that foundational work is just recognition. You're not going to remember, but when you start doing the work, it won't be the first time you've seen that stuff. This is the big piece right here. So this is really interesting because if you think about an assignment that you do in a class as something that you get done and you turn in and you get points, you may or may not be able to actually speak with other people about it in any sort of intelligent way. And what I want is I want for you to be able to talk. I want for you today, at the end of the day, to be able to talk about some stuff. I want to be able to have a conversation with you. The assessment, as it were, for this is going to be that when I come up to you and I say, well, you know, why are ribosomes so important for a cell? I don't want you to say, I don't know. And then have me say, well, do you know what a ribosome is? No. Well, like, did you do the assignment yesterday? Oh yeah, I did the assignment. I turned it in, I got the points. But you don't know what a ribosome is? Nope, no idea. That's not really learning. You're not really thinking. You just wrote some stuff down, turned it in, and got some points. What I want you to do is I want you to be able to say, okay, well, yeah, ribosomes are responsible for making proteins. And then I can say something like, great, how do they work? And you're going to say, I have no idea, man. Like, we have only looked at this for one day. That's an intelligent conversation. And in that intelligent conversation, I found out two things. One is I found out what you know, which is that you know ribosomes make proteins. And then the other thing is I found out the limits of your understanding, which is that you understand that they make proteins, but you don't understand how that actually happens, which is great for me to know, because then I know what I can help you learn next. And it's great for you to know too, because then it's going to help you realize, like, well, if I already know that ribosomes make proteins, I'm not going to spend any more time thinking about that. I don't need to. Before I smash them with a hammer. And I wouldn't actually smash the earbuds with a hammer, just so you know. I do have a hammer, and I have smashed things. I wouldn't smash an earbud with a hammer. I might smash an earbud with a hammer. I might actually smash an earbud with a hammer. I might do this right now. That'd be kind of fun. Don't you think? Can I add an earbud in here? So many other things that I've smashed over the years. You really want to smash an earbud right now. Someday you all should come up and look at my, like, what's in these drawers, because it's so fun. I have a little of everything in here. And I do have earbuds somewhere, but I just don't think I'm going to be able to find them right now. So I'll smash you later. All right. Okay, so this intelligent conversation thing, that's my goal for you. That's what I want you to be able to do. It's really different from getting the work done and turning it in. Okay, so let's think about that in the context of what you talked about yesterday. What's a cell? Mr. Fiskin explained, cells are the basic unit of living organisms, right? Like, that's, like, we all know that. You don't even have to write that down, by the way. And here's why you don't have to write that down. What does that mean? What does that mean? Anybody? This is true. And it's important to, like, know that. But what's really important is that you understand what cells are, because once you understand what cells are, then you can understand why it is that the fact that they're the basic unit of living organisms is important. Here's what a cell is. Are you ready? A cell is a package. It's a package that is made out of biochemical compounds. We talked about those. Those are, in general, lipids, proteins, carbohydrates, nucleic acids. The package itself is mostly made out of proteins, lipids, and carbohydrates, okay? You can think of it like a plastic bag, like this. It's a container. That's it. That's what a cell is. It's a container. Except that's not really what it is, of course, because even though it's a container, what's inside is super important. So the outside is made out of biochemical compounds. These are all cells. This right here is the outline of one cell. What we're looking at there, what we're seeing is something called a cell wall. And pressed up against that cell wall is something that we probably can't really see here called a cell membrane. That's the plastic bag. That's the outside of the cell. And but what's really important is inside the cell. So if outside of the cell is made up of biological compounds, what do you think is inside the cell? What do you think cells contain? What do they have in them? Anybody have any guesses? What? Say it.
Living organisms.
They might have living organisms inside of them, right? But that's not, so that's a great example of like, well, we had some definitions, but cells don't contain living organisms. Cells are the basic unit of living organisms. So we can have organisms that are made out of one cell. They don't contain a living organism, they are one. Or we can have organisms that are built of multiple cells. They don't contain a living organism. They put together make a living organism. What's inside them? What do you think, PMAC?
Chemical compounds.
Biochemical compounds? So they're made out of biochemical compounds, and inside they have biochemical compounds. Yeah, it's all the same stuff. It's not the same stuff in the sense that the specific ingredients on the outside of the cell aren't exactly the same as the ingredients on the inside of the cell. But they're the same in the sense that it's just more lipids and carbohydrates and proteins and nucleic acid. It's all the same stuff. And in fact, the pieces that make up the majority of the outside of the cell, which is something called a phospholipid, is found in abundance in the inside of cells as well. So, like if you were to take a cell and take a piece of the outside and kind of mix it up so it was all broken up, and you took some of the inside and you mixed it up so that it was all broken up, you wouldn't be able to tell if you were looking at the inside or the outside necessarily. It's the same kind of stuff, right? So this would be like a plastic bag, and inside are other things that have like outsides, right? Yeah, it's not apple, but you know, you get the idea. Maybe you pull out something like this, and so you've got these two things in there. And these things have their own outsides and insides. Cells are kind of like that. Cells are packages that contain stuff, and some of the stuff inside of a cell is just other packages. So this is what it looks like if you kind of zoom in. So here's a cell, and What we're seeing here is just some of the stuff that's inside the cell. Outside of the cell is kind of a bag that holds everything. Inside of the cell is the stuff that's all held together. So, I mean, like this is like the basic unit of living organisms. How does that work? The way it works is that it all has to do with chemical reactions. That's it. It's all we care about. Living organisms can be explained entirely in terms of chemical reactions. Those chemical reactions can be entirely explained by what is inside cells and where it's located. That's it. It's all we care about. What is it and where is it? That determines what it does and kind of when and how it happens. It's super complicated, right? So if we were to look, for example, at this, sorry, I don't know how to erase that. If we were to look at this is a electron microscope photograph of a bacterial cell. Where's the brain, by the way? Where's the brain in this, do you think? No brain. There's no brain. This is a single cell. It takes billions of neurons to make a brain. This is one cell, it's not even a neuron. There's no brain in these things. But this is, in and of itself, incredibly complex. If we were to look inside a bacterial cell, like this, what we'd see is like some of these things kind of go all the way through into the interior of the cell. There's all sorts of crazy stuff inside the cell. There's a piece that sticks out of the cell that we call a flagellum. We think of it as like a tail, but of course it's not a tail because tails are made up of like multiple cells. This is just an extension of protein and carbohydrate that sticks out of the cell. These flagella, by the way, have little motors, biological motors that spin. This flagellum spins around in circles, all inside one cell. All the pieces are made of protein, carbohydrate, lipids, mostly that. So this is what cells are. In order to understand how cells work, what we have to do is dive in here and think about what's going on. What do these little packages do? What does this do inside of a cell? What does this do inside of a cell? And we have to think about the things that aren't packaged. There's like this in a real cell. This bag would be full of stuff, just like goo. That's what fills up cells. So that's what we're going to be looking at. We are going to focus mostly on a kind of cells called eukaryotic cells. Prokaryotic cells are real simple, like bacteria. We're just not going to even focus on those. Eukaryotic cells are found in animals, in plants, fungi, and protists. And those are the ones we're going to look mostly at animals and plants because those are the easy ones to think about. But those kinds of organisms have specialized things inside the cells that have their own packaging, and those are called organelles. So eukaryotic cells have structures within them with specialized functions, and these are called organelles. Okay? And this is primarily what we're going to be looking at today. You're going to be doing most of the work. I'm going to do a very quick overview of a bunch of things, and I'm just going to throw out things for you to think about as we're looking at these. But I'm not going to ask you to write anything down, nor am I going to ask you to memorize stuff. You're just not going to be able to. It's going to be way too much, okay? So these are the kinds of things that we find inside a cell, the Golgi apparatus and mitochondria and centrioles and the plasma membrane. And I'm just going to kind of show you a few of those things because what I want is to have you see the terms, think about like what they mean, but more importantly, I want to kind of pique your interest. I want you to think about how they work and like the mysteries, the things that we don't really understand. Okay, so you do not have to write anything down. From here, I'm going to go fast. I just want you to just listen and think. So the first piece is the cell membrane. And what that does is it encloses the cell contents, and things can move in and out of the cell membrane. And that makes sense because if you think about like even a bacteria, a single-celled organism, it has to be able to get food in and waste out. So things go in and things go out of a cell. Some things can move in and out freely. Some things are like like regulated by the cell itself. It's made up primarily of those things that I mentioned called phospholipids. That's these little things that look like balloons with two strings on them. And there's also a bunch of other stuff. There's proteins and there's sugars that are kind of embedded, sandwiched in the cell. And there's also like fats, like things called steroids and stuff that create a fluid structure. So all those things together are what we call the cell membrane. And here's what I want you to think about with the cell membrane. How in the world does that work? How do you have a package that can let some things in and some things out freely? Some things are sort of controlled. They can come in but only when the cell wants them, or they can leave but only when the cell wants them to. And some things can't come in and out at all. How do you do that? Do we have anything that we use on a regular basis that works like that? Yeah. A strainer, yeah. So a strainer is a great example. It's something that can hold large particles, but not small things or liquid things. So yeah, you'd use a strainer. And that's kind of like, cell membrane is a little bit like a strainer, only it's a strainer that largely holds water. So it holds a liquid into. Other things can go in and out. If you look at a eukaryotic cell, on the inside of a stained eukaryotic cell, you will likely see kind of a darkish spot. That's called the nucleus. Inside the nucleus is an even darker spot. So this is a nucleus right here. This whole thing is a nucleus. This super dark spot right here is called the nucleolus. And what that structure is holding is what's important. It's got the DNA in it, and we'll talk about DNA more. DNA is where all the genetic information is stored. So inside the cell is another structure, and inside that structure is the DNA. Okay. And effectively, what this does is it protects DNA. So like if whatever was inside this orange was super important to me, like the orange itself, then having the orange peel around it is kind of useful, right? Because now the inside of the orange won't get damaged. There's a couple things I want you to think about though. One is, if we've got DNA packaged up like an orange, then when cells divide, which we'll talk about, somehow the orange peel has to go away, and the DNA has to get split up, and then we have to make new orange peel. So it's a little bit more dynamic than just a bag. It's like a magic dissolving bag that holds DNA, dissolves when it needs to, and reforms when it needs to. And the other piece that's really kind of interesting is if we've got the DNA protected, then how does it get used? Like how do our cells access the DNA if it's in a package that keeps it protected? We'll think about that. Another organelle, this is actually technically not considered an organelle. Organelles have to be bound by membranes. Okay, so the nucleus is bound by a membrane. The cell membrane itself is often considered an organelle because it's a membrane. The cytoplasm is generally considered a cellular structure or part of a cell. It's not an organelle in and of itself. And the cytoplasm is just everything in the cell. It's mostly this sort of gelatinous stuff. So a lot of people think about cells as being like water, like they're filled with water. And that's because they have so much water in them. But it's more like they're filled with like yogurt, right? It's watery, it's squishy, but it's more like a gel, right? You could, like, it doesn't run and flow like water does. And technically the cytoplasm has everything except the nucleus in it. And so it's, like I said, it's not technically an organelle itself. And here's what I want you to think about with the cytosol. Imagine if you had a bag filled with water, or even filled with something like yogurt, and you were to hold that bag up, everything in there besides the yogurt would just sink down to the bottom, right? Like if I put a can of soup into a bag full of yogurt, the soup is just gonna fall to the bottom. Why is it that in cells, all this stuff doesn't just fall to the bottom of the cell? And, you know, like yogurt, okay, we can kind of imagine, but, you know, cells are a little bit runnier than yogurt. And like even yogurt, if you put something really heavy in, it's just gonna sink down. So what's the deal with that? How does that work? The next thing we're going to look at is mitochondria. And mitochondria are super fun because you all know what they do, right? What are mitochondria? The powerhouse of the cell, which is actually reasonably accurate, but it's not accurate for the reason that most people think it is. Most people think about a powerhouse as a place where power is made. That's where energy is like created. Without the powerhouse, there's no energy. But it turns out that mitochondria, like a real powerhouse, does not create energy. What it does is it takes one kind of energy and turns it into another kind. That's the same thing that our power plants do. If you were to look at a hydroelectric dam, for example, it does not create power from nothing. It takes hydraulic power. It takes pressure. It takes effectively gravitational energy, and it turns it into electrical power. Why is that? Well, because it wouldn't be super helpful for our watches or our phones to be run by tiny little waterfalls. They could, theoretically, we could design a phone that would run with a water wheel. And we could make a little waterfall, and I could like have my phone, like the little wheel spins, and it's like make stuff happen in my phone. That would be ridiculous. And as it turns out, in mitochondria, things work the same way. The energy that mitochondria starts with is in the form of a chemical called glucose. Glucose is awesome, but it's not that useful for the little structures inside cells. They can't use it directly. Instead, they use something different, smaller, called ATP. It's kind of like the difference between gasoline and batteries, right? We use gas in our car, it's really useful in the car. And we use batteries in our phones, it's really useful in our phones. That is energy, they're both energy, but they have different properties and they're useful in different ways. So we don't have gas-powered watches. We do now have battery-powered cars, but as you know, until like 10 or 15 years ago, that was impractical. So that's why we started with gasoline. The next structure we'll look at is called ribosomes. So these are made up, as everything is, of biochemical compounds. And what they do is they build proteins. So remember when we talked about proteins, and Mr. Barron and I talked about how they are made up of little pieces, monomers called amino acids that are hooked together. Ribosomes do that hooking together. And they are found throughout cells. So our cells are full of like free-floating ribosomes and make proteins. They're also attached to a particular structure in the cell called the endoplasmic reticulum. And the endoplasmic reticulum is how we get information from DNA inside the nucleus to the rest of the cell. The endoplasmic reticulum is a way that we move things around in a cell. If you look at a cell's nucleus, what you'll see is the endoplasmic reticulum is connected to the nucleus. It's kind of an extension of it. And so stuff can move from the nucleus into the endoplasmic reticulum. The endoplasmic reticulum has two parts. One part just moves things around, and that is called smooth ER, because when you look at it in a microscope, it looks smooth. The other kind is called rough ER. Rough ER has ribosomes stuck in it, so protein synthesis can happen right here. Okay? So it's all like a lot. Remember, the point here is not for you to memorize this stuff. We're just building a foundation of things that you'll recognize. The next organelle is called the Golgi apparatus. What this does is it does effectively protein processing. So if you're interested in music or photography, you've done processing. If you're interested in video, you've done processing. You start with a raw material. You might start with like 3 hours of video, and you edit it down to 30 minutes. That's effectively what the Golgi apparatus does. It takes proteins and it modifies them, and it makes them work better. And then it packages them up and it ships them out. It ships them out within a cell, and in multicellular organisms like us, it can ship proteins out of our cells for use by other cells or other parts of our body. So it can send things to other cells, or it can ship things around within a cell. Some structures in cells are just sort of like storage. Like they in and of itself, they may not have a huge purpose. Many of those storage cells are designed to break things down over time, like a wastebasket. So a particular kind of storage cell is something, or storage organelle is called a lysosome. And what they do is they just collect things and break them down into pieces. They react with things that the cell doesn't need. They envelop them and bring them inside, and then they break them into little pieces and send them out. There's another kind of storage cell, and that's called a vacuole. Vacuoles in all types of organisms can be used for just general storage. They can store water, food, or waste. In cells, the vacuole can fill up with water, and that's what makes the leaves not wilt. So if you have a plant that you haven't watered for a while and the leaves are kind of droopy and you water it, that water goes into a big vacuole in the middle of the plant cell. And in different organisms, the vacuoles can do all sorts of different things. So there's some like very specialized vacuoles for different organisms. So there are more. This isn't all the organelles. These are all the ones that I'm going to talk about. And what I'm going to ask you all to do in a few minutes is to do a little bit of research into at least a good chunk of these. Before we do that, let's take a quick break. So take 5, be back in here at 2:37 on this clock, and we'll pick up where we are leaving off.